Well, hello to those of you who have entered the webinar. I'm Joy Cardine. Happy to have you with us for this. This is the first Plato-Madison lecture of the 2022-23 season. As you know, we are still virtual, but I think that's great. We can reach a lot of you this way. We'll get started in just a few minutes talking today with Dr. Sterling Johnson, and we'll learn about some of the most recent discoveries from the Wisconsin Registry for Alzheimer's Prevention, or WRAP study. We will be taking your questions for Dr. Johnson after his presentation. If you have a question, please type it in the Q&A section. I'll be monitoring that, and I'll read your question to Dr. Johnson after his presentation. If you'd like to, feel free to maybe use the chat to tell us who you are, where you're from. Maybe if you've got a special interest in learning about Alzheimer's disease and what that is. Go ahead and tell us why this issue matters to you. And I see we're already getting questions, so we'll be sure to present those after the presentation. Yeah, and we are hearing from some attendees. Actually, we're hearing from our lecture organizer and our web facilitator, Trish, who does have a very, very keen interest in this topic. Her father had Alzheimer's, and she carries one copy of the APO4 gene. So we will, I hope, learn more about that in this hour. Stacy says that she's from Wisconsin. Her mom passed away 2 weeks ago from this awful disease. Very sorry for your loss, Stacy. She battled it for 4 years and was only 69 when she passed. And really is a tragic, horrible disease. I do not have I do have an aunt who had Alzheimer's disease. I do not have a parent or a closer relative than that. My husband has Alzheimer's, Nancy says. We're from Shawano. And he is in the ANDI or Andy study at Madison, which I take it is different than WRAP. But we'll learn more about that too. Oh, Dr. Sterling's going to already answer that question. Good.
That's the ADNI study, the Alzheimer's Disease Neuroimaging Initiative.
Okay.
That's a study that we're participating here in Madison, and I'm very familiar with that study, having been the person on our campus that runs that study for the last 15 years.
Oh, okay. All right, we'll get started in just we'll give it one more minute to let some more people in from the waiting room. All right, we'll get started. Hello and welcome to the first Plato-Madison virtual lecture of the 2022-23 season, which will be on the latest research from the Wisconsin Registry for Alzheimer's Prevention Study. I'm Joy Cardine. I'm a Plato member. I'm a retired Wisconsin Public Radio host, and I'm your moderator for today's lecture. My colleague, Trish Yacarino, set up this program and is our webinar facilitator. We are recording the lecture, and it will be posted on the Plato website for those who can't be with us today. We will also be taking your questions for our guest speaker, so please type your questions in the Q&A section, and I will ask them at the conclusion of the lecture. Our guest lecturer today is Dr. Sterling Johnson, PhD. He is the Jean R. Finley Professor of Geriatrics and Dementia in the University of Wisconsin School of Medicine and Public Health. He is the Associate Director and Biomarker Core Leader of the Wisconsin Alzheimer's Disease Research Center, and he is the Principal Investigator of the Wisconsin Registry for Alzheimer's Prevention, or WRAP, which is a long-running, observational, longitudinal cohort study of 1,700 adults who have a parental family history of Alzheimer's disease. Dr. Johnson, thank you so much for being with us and for sharing some of the latest discoveries from this important study.
Thank you, Joy, and thank you to the Plato organization for inviting me to give this lecture today. I'm honored to be here and excited to share with you all some of the latest advances in Alzheimer's disease. And some of those advances are coming right through Wisconsin, and we're excited about that. And I'll convey also today my excitement for what's happening in our field. It's really a dynamic time, and there's some incredibly important things happening that I hope we'll get to talk about today. So the title of my talk is here. I'm really focused on the WRAP study, but again, I'll sprinkle in as much as I can about what's happening in our field along the way. And here we go. Let me make sure I can, there we go. So those are my disclosures. I'm a consultant to a few pharmaceutical companies, largely because we have information that's going to help them help us have better therapies for Alzheimer's disease, and I think that's a good thing. Alzheimer's disease is it's complex to think about and to explain. So I'm going to try to first explain what this disease is, the way that we think about it as researchers anyway, and give you some examples of what it looks like from a brain scan perspective or a biomarker perspective. And then we'll dig into some of the research on early detection and the early brain changes that happen. And when you think of Alzheimer's, you think of maybe a loved one who has the dementia phase of Alzheimer's disease, where they're no longer the person that they used to be. But Alzheimer's disease it's actually a proteinopathy. It means that there's some proteins in the brain that are not processed correctly, and they accumulate, they build up. One of those proteins is called amyloid, or beta amyloid to be more specific. And the other is called tau. And the amyloid clumps together outside the brain cells to form plaques, amyloid plaques. And the tau builds up inside the brain cells to form neurofibrillary tangles. You may have heard of the plaques and tangles of Alzheimer's disease. That's how we define it as researchers, is are plaques and tangles present? If so, it's, the person has Alzheimer's disease. Now, whether or not they have symptoms, that's another matter. And that's a really exciting area of our field because there's some people who can live with this disease in their brain for many, many years who don't have symptoms. And the burning question is, will they continue to live their whole life that way? Will they get symptoms eventually? What is it about them that makes them resistant to symptoms, even though they have the proteins in their brain? And then, of course, we know if we've known anybody who has dementia, they likely had dementia due to Alzheimer's disease, since it's the most common form of dementia. That's the usual course, that when you have these proteins in your brain, you go on eventually to get the symptoms, mild cognitive impairment, and then dementia. So that's already unpacking a lot of things here. So I've addressed that first point. AD is defined by its dual proteinopathy. And the second point here is that AD has a prolonged pre-symptomatic stage. And we're not good yet at predicting who's going to go on to get symptoms and when, but we're getting better, and I'll explain that. We have PET and CSF biomarkers. PET is an imaging technique. It's called positron emission tomography. PET is the short name, and CSF is the cerebral spinal fluid that we can get out of a person's lower back. It's the fluid that bathes the brain, and it has these proteins that we're talking about in that fluid. So that's one quick way of learning whether these proteins are present. And I'll show you examples of these things. Importantly, on this fourth big bullet here, other diseases may mimic or co-occur with Alzheimer's disease symptoms. And sometimes we are fooled as clinicians as to what disease may be present. If it's a progressive memory disorder, we sort of automatically think of Alzheimer's disease, but that's not always what is causing their progressive memory disorder. It might be another neurodegenerative disease. And finally, this bottom point that AD affects 5.8 million, this number is already out of date. I think it's 6.2 million now in the US is our estimate. But these are all rough estimates because they're not necessarily informed by those careful individual studies of who has plaques and tangles in their brain. They're sort of estimates based on epidemiologic studies. Well, the way we define Alzheimer's disease biologically has been come to be known as the NIA-AA research framework, and that's the National Institute of Aging and the Alzheimer's Association, two joint foundations, one governmental and the Alzheimer's Association, a private foundation. They got together and came up with this framework, and you see lots of A's and T's and N's here on the left, and that stands for amyloid, tau, and neurodegeneration. And those are the hallmark features. Is amyloid present? Yes or no? Is tau present? Yes or no? And is some evidence of neurodegeneration present? Yes or no? If a person has at least amyloid present, then they're in the Alzheimer's continuum. If they have both amyloid and tau present, they have Alzheimer's disease. And if they have neurodegeneration present on top of that, that's further evidence that that this disease is causing damage to that person's brain. Now, you can have neurodegeneration without amyloid and tau, and that might mean you have a different proteinopathy. And that's a very plausible thing. As we get better at understanding who has Alzheimer's disease, we're also becoming more aware of who has other proteinopathies in their brain. I hope this is making sense so far. Let's just dig into Alzheimer's disease, where we start with the plaques and then the tangles. And I'm showing you example images of people who have plaques and tangles here. And then brain cell death, which we can see from MRI scans. Finally, a person's brain has gone so far that we get mild symptoms and eventual dementia. This process can take anywhere from 10 to 25, 30 years or so to reach the dementia stage. So if it takes 25 years, that means that a person is living with this disease longer in this pre-symptomatic, pre-dementia timeframe than they'll live with the disease once they develop dementia, which is usually anywhere between 5 and 15 years. It's really an extraordinarily long disease, and at least half of it is pre-symptomatic. This is how we detected Alzheimer's in the old days. It's still how we detect Alzheimer's disease. These slides in the middle are slides of a person's brain after they passed away, and that's still the gold standard way of identifying this disease. But I'm showing you these examples on the left and right of an amyloid scan and a tau scan. And anything in red are areas where the scan has picked up amyloid proteins or tau proteins, as the case may be. And that means we no longer need to wait till a person passes away to learn if they have Alzheimer's disease. Here's how easy and straightforward this can be. You see two participants here, and on the top you see their amyloid scans of participant 1 and participant 2, and on the bottom you see their tau scans. And this one on the upper left, participant 1, is A positive, amyloid positive, so they would have an A plus, and it's and so that means they have amyloid present. And the participant number 2 here, who also has the same kind of memory symptoms, is amyloid negative. They're A negative. They don't have any amyloid in their brain. On the bottom, we see tau scans from these same 2 people. Again, this first participant has tau present in their brain, and which you can see here in these red crescent-shaped areas in the bottom part of their brain. This person on the right has no tau present in their brain. So there's something else explaining symptoms for this person on the right. And that means we've helped narrow it down, but we have more work to do. Okay, well, here's all the potential biomarkers. I've listed these out by their categories: for amyloid in the upper left, for tau in the lower left. These are mostly different kinds of PET scans, but we can also detect this out of the spinal fluid. There's different proteins in the spinal fluid that indicate whether a person has amyloid and tau present. And then over on the right, I've indicated and listed a few candidate markers of neurodegeneration. And MRI is the best one we have. There's also other types of PET scans that could be used. One is called fluorodeoxyglucose PET, and there's another experimental one. And there's definitely proteins in the spinal fluid or CSF that can tell us if neurodegeneration is present. But that's just to give you an idea that we've really made a lot of progress. A decade ago, there was basically one PET scan available for any of this, and there were no PET scans available for tau, and now we've got several options. And this is just the extraordinary change that's happened in our field over the last decade. And here we have just some examples of these different PET scans for tau, those neurofibrillary tangles. We see what it looks like in a group of symptomatic people here in the lower left. For one tau tracer, here's another kind of tau tracer. These are all ways of imaging the tau proteins in the brain. The point of this slide is mainly to show you that most of these techniques for scanning the brain tell us consistent results across studies, and that's good. So what we've learned so far in this, I'm going through this fairly quickly, but what we've learned so far is that AD, Alzheimer's disease, is a long-running disease. It's a chronic disease. Most of the time, it'll be a person lives more of their life without symptoms than they do with symptoms if they're destined to get symptoms. We have been studying this disease, especially this preclinical phase of the disease prior to dementia, in the WRAP study, the Wisconsin Registry for Alzheimer's Prevention. The study's been going for over 20 years. It started in November of 2001. And it was started by my colleague, Mark Sager, who was a visionary geriatrician here at UW. And he retired in 2014. And I was lucky enough to take over the study at that point. And it's just been a rewarding thing for me to do, and a study in a field of inquiry where we're making an impact on Alzheimer's disease. So our major goals in WRAP are to determine when is cognitive decline more than normal aging. And again, we're talking pre-dementia, before a person has any kind of clinical inkling that they might have something wrong. We can detect cognitive decline. And then we want to know, what are the health factors, lifestyle factors, genetic factors that might make a person more or less susceptible to this disease. We are starting in midlife, which is the sweet spot, we think, for understanding how this disease starts. And we're using all of these imaging and fluid biomarkers and technology that whatever we have available to us to understand how the brain is changing. Well, let me give you a little bigger picture of the UW. RAP is one of the projects happening here in the UW-Madison Alzheimer's disease program. We have an Alzheimer's Research Center, which supports infrastructure for research. We have an Alzheimer's Institute, which supports training and outreach to physicians and communities. These are the same people. We work in a coordinated way across these two missions of our program. And the RAP study is here. It's this cohort of 1,700 people, about 1,200 are active at any given time. People move away, or they pass away, or they age out of the study. And so about 1,200 are active. The Alzheimer's Center also has a group of people who we follow actively, and about 650 or so people are coming in year after year in our ADRC, or Alzheimer's Research Center. These two data sets, by the way, are shared interactively between our investigators, and it's a good way of identifying a finding and then trying to confirm it in the other cohort. It's a really convenient and powerful approach to doing science. So RAP is two things. First of all, it's a longitudinal observational study in its own right. And again, we started this in midlife, and we're following people over time. About 73% had a parent with Alzheimer's disease, and about 41% of our participants have this ApoE gene, which is a susceptibility gene for Alzheimer's dementia. It doesn't mean you're going to get Alzheimer's dementia if you have this gene. And also, if you don't have it, it doesn't mean you're not going to get Alzheimer's disease. It's simply a risk factor gene, and it's an important one for us. Also, RAP functions as a registry for a multitude of linked studies here at UW, where many of our professors and researchers can use RAP as a resource to answer very specific questions. And our participants are so generous with their time. They're used to getting phone calls for linked studies that says, hey, by the way, will you come down and do this study with us? And oftentimes, whenever they can, they do that. And that's just a testament to the generosity and commitment of our research participants. I've mentioned our goals are already identifying this disease before its symptoms as confidently as we can, and identifying the health and lifestyle factors that might give us a clue as to what we can do to prevent this disease. Here's where our participants live. They're all across our state and have spread all over the country as life takes them elsewhere. Some of the features of the participants are here on the left. The mean age is 67, 70% women, 73% have this parental family history. Similar to the rest of Wisconsin in this age range, 40% have a body mass index over 29, which is the definition of obesity. 11 years in the study on average for our participants. 40% have high blood pressure, 10% are diabetic, 41% have this ApoE gene. It's a risk factor gene for Alzheimer's, and about 50% have high cholesterol. Again, that's pretty common for this age range. Okay, here's some things that we've discovered that I want to go into some detail about, especially number one here. We've been able to determine the age that Alzheimer's disease starts on an individual basis. And with that knowledge, we've been able to identify subtle cognitive decline and when that is onset relative to a person's Alzheimer's disease. And we've started to try to explain and understand how health and lifestyle might slow this disease down. And our frontier is to see how we can expand that interval of time between when this disease sets in, when amyloid is first present in the brain, and the eventual symptoms. We want to make that interval as long as possible to give us enough time to intervene, to do something about it, and perhaps lower amyloid out of the brain. Okay, let me give you some examples of what is happening in the RAP study. And the first one is the study of just understanding if you have amyloid and tau in your brain, what's happening cognitively. And this is a study that we were able to do retrospectively with my colleague Toby Bedhauser, who's a professor here at UW and a specialist in PET imaging. And we asked the question in the first 160 people from RAP who had these images, what's going on with their cognition? Do people who have amyloid and tau in their brain differ in the way they're declining cognitively than normal aging? And we identified these groups. Most of our subjects, fortunately, were amyloid and tau negative, 124 out of a total of 167. There was a subgroup of 15 people who were amyloid positive and tau positive. And let's see what happened with these individuals. This is just to show that. Let me mention this first before we look at the result. ApoE, we've already mentioned this gene today. The people who had amyloid in their brain, 86% of them were carriers of this ApoE gene. So it's definitely associated with having amyloid. And it's an important risk factor gene. Okay, let's look at the result. Here it is. This is just the raw results where we've got cognition on the y-axis and age on the x-axis. We've got 4 groups of people here. This small green group just has a little bit of tau present, no amyloid. This gray group in the lower left has no amyloid, no tau in their brain. This blue group in the bottom right has amyloid only. This red group of 15 or so people have amyloid and tau. These are cognitive measurements, by the way. We have multiple measures per person, and each person is a line. And what you're seeing here is that these people are unambiguously declining cognitively. Whereas these other 3 groups, it's kind of almost random in some of these groups. They're not declining in any systematic way like this red group is. And this is just plotting it, and we can, I think, come to that conclusion just visually by looking at this. By the way, these triangles mean they had a clinical diagnosis of dementia at the time of the triangle, not dementia, but mild cognitive impairment at the time of this triangle. So these people are not even demented yet, but a few of them have mild impairment now that is starting to show up. This is, for the most part, pre-dementia, even pre-mild cognitive impairment trends in cognitive decline that we're observing. So this is the raw data. When you do the formal statistics, which we like to do, we see that this red group is indeed declining, and it's 3 times faster than any of these other groups. So our insight here is that among amyloid positive and tau positive, ostensibly healthy people who have Alzheimer's in their brain, their cognition is starting to decline many years prior to MCI, just this mild cognitive impairment stage. And that's important. Okay, let's go on to some more results with that study. We found that their hippocampal volume, which is a structure involved in memory, that was a little smaller as well, but they didn't differ on their obesity measures or their cholesterol measures or blood pressure, B12, glucose, anything like that. There were no meaningful differences. Well, what if we can go beyond just binarizing a person to being amyloid positive or amyloid negative? That's the next result I want to show you. Here's many of these same people, and we've been doing amyloid imaging in our cohort since 2009. So we have over a decade of images, and what we've observed is really striking. And that is, once a person becomes amyloid positive, which is defined as this blue dotted line here, these people in red are all people who have this amyloid protein in their brains. And what we're seeing in everybody is that their amyloid is kind of accumulating at the same rate. The heterogeneity here among these red people who have amyloid is the age at which they became positive, the age at which they crossed this dotted line that indicates they had enough amyloid in their brain for us to call it positive. And we've been able to fit some simple math functions to this to really show that this is increasing at the same steady rate from person to person. There's there's really healthy people in this cohort, and there's really not so healthy people in this cohort from a medical standpoint. There's diabetics, there's people who are obese, there's people who have high cholesterol, but there's also marathon runners and Mediterranean diet eaters and really health conscious individuals. Everybody's amyloid is increasing roughly at the same rate, and that's an important insight. I'm not saying that those things are not important. They are. They're not influencing the rate of amyloid accumulation. I'll get back to that story in a little bit. They are influencing when cognitive decline might be manifest, and that's still very important. Well, this idea of amyloid accumulating at the same rate, is that just a fluke that we found? Or could that be replicated in other studies? And it's been replicated in other studies. Here's our colleagues at WashU down in St. Louis. They get a very similar looking result to us and a very nice fit to their rates of change. Our colleagues over at Mayo Clinic are observing very much the same thing, using slightly different techniques, but showing the same thing. And here's our techniques applied to another data set called the Baltimore Longitudinal Study on Aging, or BLSA. And this is that ADNI study that we mentioned before we got started today, the Alzheimer's Disease Neuroimaging Initiative. We see the same thing in these big cohorts, that amyloid is increasing at the same steady rate, regardless if you're a couch potato or a marathon runner or anywhere in between. Amyloid seems to be fairly predictable. And what is interesting is, as I already alluded to, is that one of the sources of heterogeneity in this is that this disease can start anywhere between age 40 and age 90 plus. You can have an onset of amyloid happening in your brain. So this is definitely something that we need to think about when we talk about normal aging and dementia and survival rates. This disease can really happen anytime from midlife to late life. Okay, so what is it that influences that age of when a person gets amyloid in their brain? The only thing that we found, we've looked at sex, biological sex, we've looked at medical variables. The only thing that influences how early you get this disease is this ApoE gene that we talked about. And normally ApoE is present about 23 to 24% of the time in the general population. 23 to 24% of us have the ApoE4 risk factor in our genetics. If you have this risk factor, there's a trend that these people will get amyloid in their brain 8 to 10 years sooner than people who don't. And that seems to be what is driving this genetic risk factor. And we've published this result in a journal called Brain, and it's open access, and you can look at that if you want to take a look. So here's another discovery that was just published recently that I wanted to discuss with you briefly. And that is, what about reaching symptoms if you're amyloid positive? As I mentioned on the prior set of slides, we can determine an age of onset of amyloid, and that means we can also determine at any point in time how long that amyloid has been present, and that we term amyloid chronicity or amyloid duration. And in this figure, we've plotted amyloid duration for some of our participants, and on the y-axis, we've plotted their clinical dementia rating, which for most of us, fortunately, it's going to be a low rating, meaning there's no dementia present. In some people who are having symptoms, their CDR, clinical dementia rating, might be a low number, like 2 to 5. And then once you get above 5, that's considered mild dementia. Above 10 is moderate dementia, and above 15 is severe dementia. So these are some of our clinical research scales that we use. One of our important research scales that we use, the CDR. What we observed is that people reach this very mild dementia stage, on average, about 24 years after they become amyloid positive, 24 years. But we see a lot of heterogeneity. The mean here in blue doesn't really capture it very well. We see people here ahead of the curve who are reaching symptoms much earlier than this mean trend would predict. There's also people down here who've had amyloid in their brain an awful long time, and they have not reached symptoms, or they've reached symptoms much later than what the blue trend line would predict. And we have people over here like this. This is a professor, and this person is a speech pathologist, and they hung in there for 30 plus years before developing symptoms, and then eventually developed symptoms. So now we get into topics like resilience and resistance and susceptibility. What made these people more susceptible here is likely the fact that they were older to begin with when they got amyloid, and they also were observed to have more vascular disease in their brain, possibly signifying a second pathology or a second hit to their brain, causing their symptoms to develop sooner. So this is an important thing for us. We've got to capture not only the Alzheimer's disease signal that's present, but the other signal that's not Alzheimer's that could contribute to cognitive decline. Another insight I want you to be aware of from our studies is that after you've had amyloid in your brain for so long, 20 plus years, there's very few people down here who don't have tau. Most people have tau by this time. And on this x-axis, we have amyloid time. On the y-axis, we have some indicator of tau from these PET scans. And the blues are unimpaired. The yellows have mild impairment, not yet dementia. And the reds have dementia due to Alzheimer's disease in this case. And what we're seeing is that nobody survives this disease, or maybe one person survives this disease without developing tau. Tau is, it's going to happen eventually, but it's really variable. Some people develop tau at the same time that they develop amyloid, and some people develop tau much, much later. We have a lot to learn about tau with regard to when it happens with regard to amyloid. One thing we do know is that if you don't have amyloid in your brain, having tau is exceedingly rare. It's only 3% to 5% of the time, depending on how you draw your threshold. Okay, so that's hopefully some insights that are helping us understand these changes in the brain that cause Alzheimer's disease, dementia. How much tau do you need in your brain to cause symptoms? That was our next question. And can you answer this with just a simple, quick look at these tau scans? So we looked, and we, inspired by some colleagues, we looked to see if we stratified people based on whether tau was present in their hippocampus and related areas alone, this area where tau initially begins, or should we be paying attention to spread outside of the medial temporal lobe? And here's an example of a person who just has a little bit of tau present in their medial temporal lobe. Hopefully you can see my mouse, I'm circling this. And then down here at the bottom is an example of somebody who has tau in that same area, but you can see it's spread quite a bit outside of that little tiny focal area. And the question is, if it's spread like that, is cognition any worse for the wearer? And let me just show you the graphic on that for time. We did a bunch of stats, but this is the visual of that result that I just mentioned. These people in purple are the ones who have tau that is spread outside the area of origin, the hippocampus, and another area called the entorhinal cortex, and is spread outside into the cortex. And here's their cognition. You can see that it's dropping dramatically and has been dropping dramatically since midlife. It's got a little bit of a nonlinear shape to it, but this figure on the right is the group trend that summarizes all these spaghetti plots. And it's really this group in purple this time that we need to pay attention to, where their tau signal has spread outside of the medial temporal lobe, the area where the stuff commonly begins. When it spreads into the rest of the brain, that's when we got to pay attention. Okay, well, that's another insight. What about things on this list here in the middle of the screen? Do any of these things make you more at risk for Alzheimer's disease, or by living a healthy lifestyle on these factors, can you prevent Alzheimer's disease? Hypertension, your sleep adequacy, your physical activity levels, your stress levels, the neighborhood you live in, your diet, all of these things are in the media, the papers and our news feeds as having some impact on Alzheimer's disease. And I think what we've learned so far from what I've shown you is that none of these things are going to impact the speed at which amyloid develops. But what we found in another sub-study is that these things do in fact impact when cognitive decline becomes manifest. And in fact, the people who live a less healthy lifestyle are likely to decline sooner, not necessarily because their Alzheimer's disease is acting any faster, but because they have this poor lifestyle is causing vascular disease and inflammation and other things to happen in their brain that might just reduce the buffer that you have to tolerate Alzheimer's disease. And so symptoms happen sooner. That's what I think is going on there. This paper was published in a journal very recently by Carly Cody, who's a graduate student in my lab. And these are some of the figures from that journal, or from that article. And rather than going into that, let me just go into a couple more exciting things. I've mentioned vascular disease a few times. This is not published work. This is brand new stuff. We have observed that, just checking the time, we've observed that these little bright lesions in the brain, they're vascular lesions. We call them areas of white matter ischemia. Ischemia means there's just reduced blood flow to those parts of the brain. We've been able to quantify that, and here's 930 people who we've had these MRIs on, and we've quantified them over time. And these are spaghetti plots like we've looked at for amyloid and tau. Now we see this for this white matter lesion disease specifying, or that's informing us about vascular disease. And we see that these things are fairly predictable too. And we can actually get an indication of when a person's vascular disease may have started using the same kinds of computational approaches that we used for Alzheimer's biomarkers. So this is very exciting. It's going to help us understand the relationship between vascular disease and Alzheimer's disease. Does one start before the other? Does one cause the other? We can start to look at that. And we have started to look at that relative to the amount of amyloid in a person's brain. And we find that there's not really a synergistic effect. Just because you have vascular disease does not mean you're going to have Alzheimer's disease, and vice versa. Okay, some exciting things happening in our field that we have participated in the WRAP study. This is the idea that maybe we can detect this with a simple blood test. And I think those days are a reality. It's happening, that there will be a very simple blood test for Alzheimer's disease proteins. We can see this signal in the blood now. We don't have to do these expensive PET scans. And this kind of technique will be important in reaching more people and doing bigger studies that don't involve these very expensive scans. And so we had a collaboration with some colleagues in Sweden at Lund University, a professor by the name of Oscar Hansen and his team. And his lab ran these assays for us for this protein called p-tau. P-tau means phosphorylated tau at this particular site of the protein 217. And we took a bunch of WRAP participants, about 173 of them. And on average, they had about 3 blood samples over time each. So this is 500 and some samples on average, or in total. The age at their first plasma blood draw was 63. And at their most recent plasma blood draw that's in this study, at least, it was 68. And let me just show you this result. We asked a couple of questions. First of all, does this blood assay, do levels of p-tau217 in the blood have anything to do with what we see in the brain when we directly measure amyloid with PET scans? And the answer is yes. The longer you've had amyloid in your brain, the more likely you are to have elevated levels of this protein in your blood, which is extraordinary. The next question was, is this related to tau? And indeed it is. These people in purple, they're the same purple people we saw previously on a prior spaghetti plot. They have elevated levels of tau in their brain based on a PET scan. And we're seeing that these people also have elevated levels on this blood test. And we also see that the people who are amyloid positive, which we call PIB in some of these slides, those individuals who have amyloid present in their brain also have higher levels of this blood marker. And that's extraordinary. And so what's the agreement between a PET scan and this very simple blood test? The agreement's quite high. We do a technique called area under the curve, and the area under the curve is 0.92, which means there's just high overall accuracy, and that this blood test is accounting for a lot of the signal explained by these PET scans. So there's very good agreement. What about tau? That relationship is even more accurate. It's 0.96 area under the curve. And so these are kind of, this is the raw data up here, but these measures are telling us that the blood test is accurate. It tells us largely the same thing that our PET scans tell us. And that bodes well for our whole field. What about cognition? Can we predict cognition with these blood tests? Yes, we can. The people who have high levels in the blood versus medium and low levels in the blood are declining at different rates. This is now cognition on the y-axis and age on the x-axis. The people with high levels of this p-tau217 protein in their blood have been declining more rapidly compared to those other two groups of medium levels or low levels of p-tau217. And it seems to be a lagged relationship with cognition, such that the blood marker is changing first, and then the cognitive marker is changing several years later. And this slide is to show that we can take these same techniques for developing temporal models and age of onset to this blood marker. So these are the serial blood markers, serial blood measurements in the same people. Each line is a person. And we see that on average, that the rate of change seems to depend on how much is present. The more p-tau217 that's present in the blood, the faster the rate of change. And that means we can fit this kind of curve to it, and we can get an estimate of age of onset from a simple blood test, and that's extraordinary. This all needs to be confirmed. We have some stuff on this that looks like it is confirming it, and that's very cool. So in summary, with this blood data, we have what I think is a home run. We have an accurate measure in blood for amyloid and tau PET scans. It can serve as a surrogate marker for amyloid PET, because the more the PET scan is changing, the more amyloid is present on a PET scan, the more the p-tau217 blood marker is changing. Blood predicts cognitive decline, as we saw, that the people with higher levels in the blood are declining faster. And in these blood markers of p-tau217 anyway, we can get an estimate of the age that it first became positive, meaning that we'll get a referent or an anchor point to which we can measure cognitive symptoms and determine who is resilient or susceptible to cognitive decline for a given level of amyloid in their blood. So very exciting stuff. We think that going back to a slide I showed you earlier, it's so important to start surveilling the general population for these proteins. And why is that important? It's because we now have a positive clinical trial that says we can lower these proteins to normal levels in the brain. And if you were following the news a couple of weeks ago, you may have seen that there's a drug called lecanemab. It's a vaccine type of a drug for Alzheimer's, not quite a vaccine per se, but it works with the same idea in mind, that it works with our immune system to attack the plaques and pull them out of the brain. And this is so important to understand on an individual basis because of what I showed you earlier. Amyloid can start anywhere from midlife to very old age, and we won't know who has it unless we measure it. It's impractical to do this with PET scans or with lumbar punctures. We need to use blood, and that is now going to be possible in our very near future. What if we find a positive blood test? We will, I hope very soon, have an FDA approved and Medicare compensated drug that removes amyloid out of the brain. There's a lot of research being done on this, and I can talk more about that in a second, but I just thought I'd end with a case example from RAP, who is giving us reason to be optimistic and also scratch our heads a little bit. This person has a lot of amyloid in their brain and a lot of tau in their brain. When we last did their amyloid scans at age 64, she had been in the study for 10 years. She has 12 years of education, but had a baseline IQ estimated around 120, so a very bright individual. She's been able to tolerate this protein level in her brain. Her cognitive scores have been staying pretty steady. I don't know how long they'll remain steady like this, or whether we'll see a precipitous drop or a slow and steady drop. We don't know. This is the kind of this is the kind of person we want to learn from, to understand why are they resilient. What is it about them that is causing them to withstand all these proteins in their brain? So a hopeful case example, I think. Well, in summary, Alzheimer's is detectable years prior to its symptoms. We can detect this with biomarkers. People who have both amyloid and tau present have been declining from midlife based on our findings. This biological framework for amyloid, tau, and neurodegeneration works very well, but we have been able to take it even farther by showing that we can get an age of onset, and that means a disease duration that improves our ability to predict and to understand symptoms in relation to the time a person has had this disease, the amount of time they've had the disease. What influences amyloid onset age? Well, it's influenced by that gene, ApoE4, but that's not the only thing that influences it. We don't understand the other things. We can derive temporal information from a single PET scan, and I think we'll be able to do the same thing from a single blood draw using the computational models that I showed you examples from. We can get an estimate of when that disease began and therefore its duration from a simple blood draw or from one of these PET scans. Amyloid in the brain accrues very slowly and predictably, but it's not benign. The longer you've had it, the more likely you are to exhibit the other protein, tau, and also exhibit eventual cognitive decline. Lifestyle and health factors are super important. They don't stop amyloid from accruing at its inexorable pace, but they do either speed up or slow down when cognitive decline is about to happen. And also, blood markers for this disease are feasible, and they're coming to our clinics very soon. Right now, they're still experimental, but it won't be more than, I'd say, a few more months before they're available in clinics. So it's a very exciting time. We couple this with what we've learned from this recent clinical trial from lecanemab, this drug that lowers amyloid to normal levels. I think we have a future where maybe in 2023, we will have a drug approved by the FDA that is uncomplicated and clearly shown in the clinical trial to lower amyloid and to slow down cognitive decline. And if we can identify the right people to be on that kind of drug, we will be able to, I think, change the game of this disease, or change the face of this disease, and prevent more people from developing its symptoms, or at least slow it down dramatically compared to the tools that we have now. I want to thank the NIH for funding all of this, and all the people in my lab and the RAP staff who were so dedicated, and the participants, and especially these people listed by name here on the right, who have helped make the slides possible that I showed you today, and the research possible. So let me stop there and answer any questions you have.
All right, wonderful. If you have a question for Dr. Johnson, you can post it in the Q&A section, and we will present those questions to him. I'm going to start with a question that was posted before we even started, and you touched on it in the presentation. Talk a little bit more on this Alzheimer's vaccine that you referred to. The participant wanted to know if we're going to have an Alzheimer's vaccine anytime soon.
Yeah, it won't be a vaccine that you can get in your 20s and it'll prevent it from happening in your 80s. But it's an immunotherapy. It uses vaccine style technology, immune technology, but we give it through an intravenous infusion, and it goes and finds and potentiates the immune system to find those plaques and to break them down using the body's own immune defenses. And so that's where we call it a vaccine. It's not a true vaccine. It's an immunotherapy. And that drug was called lecanemab by companies Eisai and Biogen, and it had a positive clinical trial announced about 3 weeks ago. They'll be presenting more of that work later this fall at a conference and then publishing their work, but they just sent out that press release showing that this immunotherapy drug reduced cognitive decline by 27%. So it slowed down cognitive decline by 27%. And it also lowered amyloid to normal levels over a span of 6 to 9 months. It lowered amyloid to normal levels, and the cognitive change was over a 18-month span. And it's an incredible finding for our field. It's the first disease-modifying, positive, unambiguously positive clinical trial that we've had. There was another positive trial about 1.5 years ago called aducanumab by these same two drug makers, Eisai and Biogen. That one was stopped in the middle, but it did have a positive finding among people who had a high dose of the drug for a sufficient amount of time, and they got FDA approval for it, but there was enough of a backlash with that it just was not successful. This drug has fewer side effects, this new drug, fewer side effects, has greater efficacy in reducing amyloid, and has been shown unambiguously to reduce cognitive decline. So it's a good thing.
It's amazing. It really is. Very exciting. You had mentioned at the conclusion that we need to ID the right kind of people to take the drug approved by the FDA in 2023. Yeah. How are you going to do that?
You got to find the right people for the right drug at the right time in life, is what I meant to say there. And that is the right people would be the ones who have amyloid in their brain. And the way we would do that, I think, is by blood screening. People who are interested would get a blood screening test to see if they have the proteins in their blood, and then be invited into a clinical trial. And those kind of trials are happening now. That technique is proving very effective. It means we don't have to have people in these fancy PET scans that are difficult to do and take a lot of logistics and they're burdensome. We can do it with a simple blood test. So that's how we'll get there.
One of our attendees asks, what age is the oldest at which you would accept volunteers? Now, I'm guessing that's for the RAP study overall, or maybe clinical trials. But talk just a little bit on whether you have age requirements or what are the requirements.
Right. Yeah. For the RAP study, it's age 40 to 65 in general is when we want to enroll people. We want to capture that midlife time span. We're enrolling people who come from either a rural background or from an underrepresented demographic group right now. And for participants, for people listening to this who are either from a rural address or from an underrepresented group in research, being Black or Hispanic or Asian, these are groups that we're trying to now round out our study with so that we can improve the diversity and generalizability of the findings.
Another participant asks, when a blood test becomes available, at what age would you like to see it used?
I think it's going to be more or less like colonoscopies are done every 5 or 10 years, starting in midlife, starting perhaps at age 50, and then again at age 60, and then at 65, and so forth. We'll come up with some sort of recommendation like that. There'll be an appropriate use panel that figures all that out, but it will be feasible, I think, within a few years to have that kind of population screening happening.
Good. Nicole says, my mother just passed at age 69 from Alzheimer's. What age should I get my blood tested?
Yeah, sorry to hear that, Nicole. It's when these tests are clinically available, that would be the time to do it. They're not yet clinically available, so it's only possible in research right now. And this disease, you may not ever get this disease. We know from family history studies like we've done that only 25% of our adult children have the proteins in their brain. So 75% of people with a parent who had AD are off the hook. They're not going to get it. We don't know which one you are, Nicole, until you get a blood test or some biomarker test in research. Now, if a person has symptoms, I should just point this out, that they should see their doctor. They shouldn't wait for this technology to evolve. They should go and see their doctor now and get evaluated now. And there are some of these blood tests that are available. There's also some of these other biomarkers available now that your clinician may be able to hook you up with.
Is the RAP study still recruiting participants?
It's, as I mentioned a second ago, it's just recruiting people from certain demographic groups, rural backgrounds, and from ethnocultural minority groups right now.
Is it possible to reverse the vascular changes seen on an MRI in the brain?
I sure hope so. We don't know that for sure, but I would like to think that that would be possible. From the plots I showed you, though, I didn't see a lot of people downturning once they had these white matter ischemic lesions in their brain. They weren't turning back down. So we need to do more work there to figure that one out.
Any clue how the ApoE gene correlates with amyloid?
Yeah, it's a risk factor for amyloid. If you have amyloid, you're going to be ApoE positive about 80% of the time. But on the other side of that, there's a lot of ApoE4 carriers that never do develop amyloid. It's just that if you do develop it, more than likely you're E4 positive, but the majority of E4 carriers don't develop amyloid.
Is there any science behind developing Alzheimer's being due to or related to cancer?
There is science that has looked at this, and there's not a relationship that we're aware of between cancer and Alzheimer's disease.
Several years ago, we read a large Finnish study concluding that regular sauna use significantly decreased dementia and heart disease. Can you comment?
It probably isn't influencing amyloid proteins, but it does shock the system and maybe is therapeutic for blood vessels. I don't know the mechanism of that, but that would be my speculation, that it would be working through a vessel vascular type of a pathway.
Yeah, I recall, again, it was older studies, but that what was good for the heart was good for the brain, and that we should just be, the exercise and diet and lifestyle things that were going to keep our heart healthy, we're going to keep our brain healthy. That doesn't sound like that's exactly the case.
It's not exactly the case, but it's still good advice. It's going to keep our brain healthier for longer. If you have Alzheimer's on board, which some of us will have that in our future, but most of us won't. But if you do have it in your future, the healthy lifestyle is going to prolong good quality of life. And we don't know how many years it will provide of quality of life. And that's something that we're actively studying. But I would definitely want to continue or urge people to continue to do what's good for the heart, because it is good for the brain. It just doesn't slow down Alzheimer's disease per se, but it does enhance the brain's health overall, and it gives us some protection against the eventual symptoms.
Another participant said that she did the 23andMe DNA testing, and I have one variant of the APOE gene, and my mother had Alzheimer's. What are my odds not considering lifestyle?
Yeah, the overall risk rate, if you're E4 positive with one allele, it's about 3 times the risk of the normal population. So our normal population is about 12%, just random people, random 65-year-olds off the street. There's about a 12% likelihood that they'll eventually get Alzheimer's disease. So if you have one copy of E4, that triples. So it's now about 36% likelihood, which phrased another way is 64% likelihood that you won't.
Yeah. So there seem to be a lot of children of parents in our audience today who had parents who passed from Alzheimer's. What general advice do you have for them? They're obviously, understandably concerned and probably frightened that this could also happen to them.
I think we have such good news coming down the line with this clinical trial that just happened. There'll be other clinical trials that look like they'll be positive too in our near future. And so I think, first of all, we want to have hope that it'll be realistic, that there'll be a treatment within the next 3 to 5 years for asymptomatic individuals. Certainly for people who are already showing symptoms, if there's any concern that it might be Alzheimer's, or if you're just wanting to get it checked out, the advice is to go and have that looked at. But for the majority of us who might have a parent with AD, I think let's stick with the healthy lifestyle habits and get involved if you want. Get involved in research. RAP is one study. There's many other studies happening in our UW program. There's prevention clinical trials, including one with this drug that we've talked about, lecanemab. Get involved with those kinds of studies, and or help in some other way that we can talk about.
Does Alzheimer's have anything to do with aluminum pots? Maybe talk some on, is this entirely genetic, or do we think there's any environmental factors involved?
That's a great question. It's probably not much to do with aluminum pots. That was an idea about 20, 30 years ago that didn't pan out,
so to speak.
so to speak. But it just shows us that what happens on the epidemiological scale does not often translate into clinical wisdom or clinical treatments. A while ago, we were told that arthritis medicines would help slow down our Alzheimer's disease, or that statins for treating cholesterol would slow down Alzheimer's disease. And those have not panned out in clinical trials. So sometimes what we observe at an epidemiologic scale doesn't really translate.
A personal question. I also did that 23andMe DNA testing, and I do not have the variant that they tested for. I don't remember the exact wording, but they said you do not have, the two markers we tested for, but that does not mean you won't get Alzheimer's. So right. I'm not out of the woods, huh?
That's right. We saw on one of those slides I showed, I was showing you E3/3 homozygotes, which is the most common form of ApoE. They don't have the risk factor gene. And even in that group, we saw that the age of onset is anywhere between 40 and 90 plus. And so, yeah, we're not out of the woods just because we're not E4 carriers. It just means that there's slightly less likelihood. The general base rate is about 12% for a 65-year-old that they'll go on to have Alzheimer's disease at some point in their future. So that's the rate for E3/3 homozygotes for ApoE.
Another attendee says, my mom is 95 with very mild cognitive decline, but on PET scan, full-blown AD. She pleases and surprises her doctor. What's going on there?
Yeah, that's an example of resilience or resistance to this disease. If she got a PET scan and it was full-blown, that's probably an amyloid scan. So we know one of the proteins. We need to know if she has the other protein, the tau protein, and that could be done with a research PET scan. That's probably what's going on, is that it's really that tau protein that's driving symptoms. Amyloid is necessary, but not the sufficient key ingredient to drive cognitive decline. And so that's probably part of it. And the other part, maybe your mom's healthy living for 95 years. Who knows? It's a lot we don't know.
Anonymous attendee says, I've been in RAP for about 19 years, so almost the whole thing. I have amyloids, but don't remember, ironically, about tau. I'd like to volunteer for a clinical trial of the new therapy. How does one go about that?
Look at the website for either for our Alzheimer's Center or for the AHEAD study, AHEAD, AHEAD. And that is the clinical prevention trial for people who are amyloid positive. And if you want to email me or any of the RAP team, we can point you in the right direction for that anonymous attendee. Thanks for being in the RAP study.
Yeah. Is there any correlation between diabetes type 1 or 2 and dementia?
That's gone back and forth. There's nothing that is strong enough that we can hang our hat on for that. But I think of what's going on there is that people with diabetes they're more likely to have vascular disease in their brain, and therefore more susceptible to cognitive decline if they also have Alzheimer's disease. But right now, there's no strong relationship between diabetes and Alzheimer's.
Are people enrolled in the study told of their test results all along the way?
No, and it's just been, I think we started telling people their cognitive results in 2009 or 10, somewhere around, maybe it was even later than that. But just recently, we've gained permission from the Research Oversight Board to share our amyloid PET results. So we can now share that with our participants who want to know their amyloid PET scan values. And we are happy to share that with them.
Well, we better wrap it up, even though we're still getting some questions here. Maybe so what is next, Dr. Johnson? What more do you want to know?
I'm just grateful to be here, and I'm really appreciate everyone attending and for your interest. If you have other questions, you can certainly email me at scj@medicine.wisc.edu. You'll find me on the wisc.edu website if you Google me there. Be happy to answer any other questions you have.
Wonderful. Thank you very, very much for being with us. Very, very interesting and fascinating. Sterling Johnson is a professor of geriatrics and dementia at the University of Wisconsin School of Medicine and Public Health, the associate director and biomarker core leader of the Wisconsin Alzheimer's Disease Research Center, and the principal investigator at WRAP, the Wisconsin Registry for Alzheimer's Prevention. And again, this webinar will be at a later date, soon be posted on the website for your viewing or reviewing pleasure. The next Plato-Madison lecture will be November 9th at 1 p.m. We will hear from a University of Wisconsin-Milwaukee professor who has authored a book on the history of soap operas. That's right, soaps. The book, Her Stories, details the cultural and economic significance of daytime television soap operas, and we'll hear all about it next time. Registration for the soap opera lecture is open on platomadison.org. On behalf of Plato Madison, I'm Joy Cardine. Thanks for joining us. Take care and keep learning.
Thank you, everyone.